Incredibly tough and flexible, while also biodegradable and biocompatible, spider silk has many potential uses. Ideally, an artificial version of the material could be mass-produced, but scientists have struggled to sufficiently replicate the complex molecular structures of natural silk that make it so unique. Now, in a major advance, a team at the Riken Center for Sustainable Resource Science in Japan has used a microfluidic device to mimic the chemical and physical processes that occur in a spider's silk gland. Reporting in Nature Communications, the group is the first to show they can make silk fibers with hierarchical structures using such a device. Another perk, by drawing on spider biology, the method also is eco-friendly and avoids harsh denaturing treatments that have previously been used to make artificial spider silks. The microfluidic device is a simple-looking chip with 3 inlets, several channels, and 1 outlet. Similar to how natural silk in spiders is pulled rather than pushed out, the device operates under negative pressure, and the artificial silk self-assembles as it travels through the chip. First, spider silk protein, or spidroin, is loaded into the device. It then mixes with a buffer to trigger liquid-liquid phase separation, creating protein droplets. The droplets then encounter a drop in pH, which triggers nanofibril formation. Traveling along the straight channel, shear stress then further promotes assembly of long fibers. The resulting fibers show a clear hierarchical structure that are highly flexible and stable. Raman measurements confirm that as the spidroin moved through the device, the artificial silk developed more beta sheet structure. Beta sheet formation, the researchers discovered, requires a certain level of negative pressure, namely shear force. They also observed increases in beta sheet content at higher pressures, suggesting pressure may be a way to fine-tune artificial silk fiber structure. Indeed, the microfluidics device offers a way to further probe the mechanisms of spider silk assembly. This will not only move the field of artificial spider silk forward, but also may shed light on how spiders naturally spin silk. Future work will investigate the self-assembly of the artificial silk when adding another key spider silk protein. The researchers also plan to swap out specific amino acids in the silk proteins to explore the critical first step of phase separation.